intestinal tract. So the part that is of interest to us is from the stomach to the rectum. Okay? We will not talk too much about the stomach, just a little bit in one of the lectures, but as you know, as you probably know from other courses, from the stomach, the more after the stomach ends into the duodenum, right, which is the more proximal region of the small intestine. So the duodenum then becomes the jejunum, right, which is the longer section within the small intestine, which ends with the ileum. And the ileum is the distal region of the small intestine, and through the ileocecal valve continues into the cecum, which is here, and which is the most proximal part of the large intestine. The cecum is relatively short and continues in the ascending colon, the transverse, the descending, the sigmoid, and the rectum, which ends with the anus. Okay? So these are the main regions in the intestinal tract. The bacteria, the microorganisms, are found everywhere along the intestinal tract, even in the stomach. Even though in very low numbers in the stomach, obviously due to the characteristics of the environment there, but a few can be found there, but most of them are found from the ileum down. Okay? And the largest community is in the colon. So that picture there is another study that shows how the gastrointestinal microbiota clusters separate from microbiotas at different regions along the body, so it's similar concept to what we saw earlier. And this is a pie chart that shows the relative abundances of the main genera of bacteria that are found within the intestinal tract. So, what is found in the gut microbiota? So, most of these studies that we see, except if it's indicated, were generated using stool as a sample. Because, as you can imagine, when you run a clinical study, so with human participants, the easiest sample that you can access in a non-invasive manner is the stool, right? So the stool is used as a proxy for what is found along the intestinal tract. But I'm sure that you already can think that it is not a great proxy, right? So most of what we see in papers is data derived from the stools. The stool is very similar, the composition of the stool is very similar to the composition of the distal colon, but it's very different from the more proximal regions. And we will see this more in detail. So just keep that in mind. It's just that it's much easier to collect a stool sample. And I can tell you it's already very difficult to convince people to give you stool samples. And, but at least it's non-invasive, right? So in a clinical study, especially nutrition studies, that's the sample that we get to work with. Okay, so this slide shows that microorganisms in the gut are very diverse. So it's not only bacteria, right? Even though most of the microorganisms are bacteria, like more than 90% is bacteria, but they're not only bacteria, there is also archaea, there's viruses, and there is eukaryotic microorganisms. So the bacteria, that is a representation of what the relative abundances are between, what the relative abundances are among different phyla of bacteria in this intestinal tract. So these are different individuals that were studied. So stool samples collected from different individuals, and then they were sequenced, and then the data were analyzed and assigned to the different taxa, and we were able to see that what the relative abundances are. So for example, the light blue is the Firmicutes, phylum Firmicutes, Bacteroidetes, actinobacteria and proteobacteria are the four main phyla that are represented in the gut microbiome. The viruses altogether refer to as the virome, right? So again, we are used to think of viruses as pathogenic viruses, right? Most of the viruses in the intestinal tract are bacterial viruses, so are like phages. And there is a virome, and we are starting to appreciate the importance of this virome, but largely understudied compared to the gut microbiota, right? The most of our knowledge still today comes from the bacteria component, and now there are more and more studies that are focusing on the virome or on the archaeome, which is the collection of all of the archaea. Right? So archaea, as opposed to the bacteria, are not present in all individuals. Or I should say, based on the sensitivity of the techniques that we are using, we have not been able to find archaea in everyone. So in this study, for example, they found up to 10% of the microbiome is archaea, but only in the archaea carrier individual. Though keep in mind that there are some people who are considered non-carrier, like they don't have archaea, but again, it may just be because we are unable to detect them. Okay, so we don't know yet. The presence of the archaea, though, it's considered to be important. So again, the archaea, I told you already, were identified as a separate domain of life only quite recently. So our knowledge of the archaea generally is limited compared to the bacteria. So of the archaea within the intestinal tract is even more limited. But we know that there are many orders that are represented. So this yellow here, so all of these are the archaea that have been found in the digestive tract. And those in yellow are the methanogens, because remember that the archaea are the ones that produce methane, right? They use the hydrogen produced by the other microorganisms and the CO2, and they produce methane. So they are the only methane producers in the intestinal tract. So those that are highlighted yellow are the methanogens, or the archaea that are able to produce methane. There are other archaea that are not able to produce methane. So as you can see, these archaea can be found along the intestinal tract, some of them even in the oral cavity, and for some of them we have their genome sequenced, so that our knowledge is advancing, but there's still a lot that we have to find. Okay, so if we go back to the bacteria, as I was saying, four main phyla, the firmicutes, and the actinobacteria are the gram-positive, and the proteobacteria and the Bacteroidetes are the gram-negative. Those that are in bold is because these are the dominant phyla. So essentially, I would say most of the microbiota is constituted of firmicutes and Bacteroidetes in different relative amounts, followed by actinobacteria and proteobacteria. Okay. I am speaking like generally speaking. Okay. Keep in mind, we will see data. There are individuals with a ratio of firmicutes to Bacteroidetes, which is very skewed towards the firmicutes or the Bacteroidetes. Right? There is always extremes. But generally speaking, those are the two main phyla, followed by actinobacteria and proteobacteria. So as you can see, at the phylum level, there is little diversity. These are not the only four, but they are the predominant. Okay? But there is little diversity at the phylum level. So where we see lots of diversity is at the lower taxonomical ranks. So even below species and strain, this is where we have like a very high diversity. So altogether, altogether, we have about 10 to the 14th microorganisms that account for about 1.5 kilograms of our body weight. Right? So their concentration, their density increases going distally. So see, only 10 to the second per gram of intestinal contents in the stomach, down to 10 to the 12 in the colon, number of cells per gram of intestinal content. So the composition also varies along the intestinal tract. And it may seem obvious, right? So obviously the environmental conditions and the nutrient supply is not the same in the stomach, in the jejunum, and in the large intestine. So depending on the environmental conditions, certain microorganisms can thrive better than others. So one easy distinction between small and large intestine is the presence of oxygen, right? So in a healthy individual, the large intestine is basically anoxic, so there is no oxygen. So anaerobes can thrive there. On the other hand, in the small intestine and in the stomach, of course, there is much more oxygen, and different kind of microorganisms live in those regions. For example, the lactobacilli, they are mainly found in the small intestine. There are lactobacilli also in the colon, but they're mainly found in the small intestine versus certain Clostridia, which are strict anaerobes who cannot live in the presence of even, or the archaea, like the archaea, cannot stand even the smallest oxygen concentration, right? So these are found in the colon. So as I was saying earlier, we use stools in many clinical studies, but the composition along the intestinal tract varies. And in this study with healthy individuals, they actually showed it. So they focused on these 6 different taxa, and they used specific probes to quantify them in the cecum versus the feces, right? So remember, the cecum is the more proximal region of the large intestine, so it follows the ileum, right? It's where the large intestine starts. And they compared the abundances of these 6 bacteria in the cecum and the feces, and the numbers were different, right? So always keep this in mind, that what we're studying and which conclusions we are drawing from those data. So, So this figure was constructed using data obtained from DNA sequencing, DNA that was prepared from stool samples of human beings, right? And this shows the abundances of various taxa and their relatedness. So again, it shows which are the main genera that are found in this fecal microbiota. So these are names that you have to remember. So there are both gram-negatives, gram-positives, as we said earlier. Among the gram-negatives, the predominant taxa are the Bacteroides and Prevotella, and the Enterobacteria. Okay. We will hear this name a lot, so you will find it easier to remember. And among the gram-positive, the Clostridia, which are the main fermenters, right, together with the Bacteroides also, the Lactobacilli, and the Bifidobacteria. So the gut microbiota has a high inter-individual variability, but there is a higher degree of similarity between family members. And then it's true that there is variability, but the question was, is there a set of microorganisms that is conserved and present across all or most individuals? So this set is what usually we refer to as the core microbiome. So this concept was introduced in 2009 by this paper, where they studied 17 different individuals, and they were able to find a certain number of operational taxonomic units which were able to distinguish their microbiomes, and among all of these OTUs, 66 were representing 2% of the total detected OTUs were present across all individuals. So the authors of this paper concluded that yes, there is a core microbiota, and that this set of genera, Faecalibacterium, Ruminococcus, Eubacterium, Doria bacteroides, Aristippus, and Bifidobacterium are present in at least 50% of the people. Right? So it's difficult to come up with a specific definition of which genera or taxa constitute the core microbiome across all individuals. Right? It's very, very difficult, and we cannot do it as of today. Probably we could do meta-analysis across data sets generated from individuals in different areas of the world, and but still it would not be like a definitive answer. Yeah.
Yeah. Then when you refer to core microbiome, does it refer to a general idea of like what is similar between multiple individuals in the study, or does it refer to those specific?
So, yes, they can only conclude for the people that they studied. Okay? So usually when you conduct a study, you would select your sample in order for it to be representative. Right? But then we can ask ourselves how representative can be a data set from 17 individuals. Right? Though we always have to keep in mind how difficult it is to conduct these studies. Obviously today it was 2009, right? So 17 seemed like a large number at the time. Today we would probably have to use hundreds to come up with a similar conclusion in order for it to be considered sound, right? So yes.
Then is 50%
of these.
That's the magic number, it has to hit 50? Thank you.
Well, it's not a written rule, yes, but they said, okay, if it's present in at least more than 50%, which for them was the 50% of 17, right? Then we consider it to be likely to be core. Yeah. No, but I agree with you. There are many people who have no bifidobacteria and they are perfectly healthy, and we have always thought historically that bifidobacteria are necessary and were the driver of intestinal health. And so there are many things that we had to learn to relearn in the field of the microbiome. So now, next to the core microbiome, another concept that was put forward is the concept of the enterotypes. Okay, so this concept was proposed in a Nature paper in 2011. So in this work, it was proposed that individuals could be classified across the world. Everyone could be classified as belonging to one of three enterotypes based on the composition of their microbiome. So they said everyone has either enterotype 1 or 2 or 3. Okay? So they said enterotype 1 is driven by Bacteroides. Enterotype 2 is driven by Protella, and enterotype 3 is driven by Ruminococcus. Right? And here they represented, so in their study, they put together data from multiple sequencing effort studies, and that's what they came up with. Like in their data sets, which was a quite large one, people would belong to one of the three. So enterotype refers not just to one genus, in this case, being present, but to the relative abundance of that genus, which for enterotypes 1 to 3, the Bacteroides was more abundant, the Prevotella for number 2, and the Ruminococcus for number 3. But also to the taxa that co-occur with this genus, right? So remember one of the first slides today when we saw those co-occurrence networks and how the microorganisms can be found together and be in relationships that are positive or negatives, right? So this is something similar that they did here. So they found that people who have a Bacteroides type of microbiota they not only have higher numbers of Bacteroides, but this Bacteroides is related with this other taxa here in either a positive blue or negative yellow correlation. Remember I told you it's not always red and green, so this is an example here they use blue and yellow. Those that have an enterotype 2 driven by Prevotella, the Prevotella is co-occurs and is correlated positively or negative with these other taxa, and the same for the type 3. Yeah.
You know how big the sample size for the study was?
Ooh, I don't remember. Let me see. I know that they had both healthy and people with inflammatory bowel disease. The sample size, it should be all these dots. So we can go to the paper and find it, but yeah, obese and IBD. Yeah, so every person is a dot. So here they are 6, maybe 30 or 40. So the structure of the microbiome would be different according to the enterotype. So obviously you don't have to remember like all these relationships, right? It's just the concept that you have to remember. So interestingly, there was a follow-up study that found that the type of enterotype is correlated with the dietary type. So they looked at long dietary pattern like in the long term. So they used FFQs for a year, I think, or even more. And they found that people with a Prevotella enterotype preferentially had what they called an agrarian carbohydrate-rich diet, while people with a Bacteroides enterotype had more of a Western diet rich in animal fat, protein, and saturated fat type of diet. So it seemed that the type of diet was a driver, or at least not a driver, that there was a relationship between the diet and the enterotype. So I note here that there is no enterotype number 3, right? So that's because as research advanced, we came to learn that maybe enterotype 3 is not that defined, and that actually enterotype 1 and 2 does not mean that people are either 1 or either 2. In fact, we became to learn that there is a gradient of people. So 1 and 2 may be like kind of extreme type of enterotypes, but in fact, we all have our own enterotype, which is in between. So the distinction may not be so drastic, and there may be more of a gray line. Now, you know, it's very difficult to know where the truth lies, because there are school of thoughts. So there is a current of thought, a line of thought of scientists, microbiome scientists, that think that no, we have enterotype 1 and 2. And then there is another line of thought that thinks that no, there is more of a gradient in between the two, right? So we will see. We will find out as the time passes and there is more research data that are generated. But lately, if we compare disease and healthy, yes, the distinctions are more clear. But when we look at healthy people, especially in relation with diet, it's probably not that easy to find like drastic differences, and it's more of a gradient that in fact exists. So that other study down there, I put it there because it was the first study to look at people with obesity and people who are lean, and to compare their gut microbiota. So this study found that the relative abundances of the Firmicutes and Bacteroidetes, so those main two phyla, were completely, the relative between these two phyla was totally different in people with obesity as compared to lean. Right? So look at the lean here. In the individual with obesity, there were much lower abundance of the Bacteroidetes and much higher abundance of the Firmicutes, so that the ratio was different from the one in lean people. And then when put on a low-calorie diet for a year, then the microbiome of the people with obesity tended to resemble more and more that of the lean individuals. So this was a pioneer study that showed how the composition of the microbiome varies with diet, in this case in response to diet, and how it's linked, it may be linked to obesity. So it opened a completely new avenue of research, that paper. So it's a really pioneer study, and we'll talk about that more. There is a lecture on the metabolic syndrome, so we'll expand on this concept.
So as of right now, we aren't really sure if these microbiomes cause the weight loss or the weight loss caused the microbiome. We aren't
Today
sure.
we don't know yet. In a few weeks we may add to that knowledge. In fact, it's probably both ways. And so yeah, that's what probably is. So I mentioned a few times that our body, to stay healthy, has to be able to handle this massive community of microorganisms that reside in our intestinal tract, right? So how does it do that? So first of all, where are they located, these microorganisms? Because we say they are inside the intestinal tract, they are in the colon, but where? So a little refresher of the anatomy of the intestinal tract. Right, so as you may remember, there is a monolayer of intestinal cells, right? The enterocytes, absorptive cells, but also immune cells. So, and these enterocytes are covered by a layer of mucus, right? So that the microorganisms are not in direct contact with our intestinal cells. Actually, it turns out that the mucus layer is constituted of two sublayers, one that is more firm and one that is more loose. Right? So the one that is in immediate contact with the intestinal cells, so this part of the mucus layer below this dotted line, is devoid of microorganisms. Right? So there are no microorganisms that are in direct contact with our intestinal cells. The more loose the outer mucus layer has actually microorganisms embedded. And then there is more on its surface and there's more in the lumen. Okay? But the microorganisms in a healthy intestinal tract do not come in direct contact with the enterocytes. Now, there are a few exceptions, like a dendritic cell that is found in the mucosa, right, can protrude in between two adjacent enterocytes and sample a bacterial cell from the lumen. Okay, so in that case, obviously there would be a direct contact with the immune cell. The cell is protruding, which is part of our system for tolerating microorganisms, right? But otherwise, there is a mucus layer that protects and creates a physical separation. So it is very important that the microorganisms that are found in the lumen stay in the lumen. They are not supposed to travel around and translocate into the circulation, because otherwise they will cause disease, right? We don't want them, to the 14 microorganisms, to cross the intestinal barrier going to the circulation. So our intestinal tract has developed this capacity so that there is intestinal barrier that seclude, like that prevent the crossing of the epithelium layer in the intestinal tract by these microorganisms. So they stay in the lumen. So this mucus layer, as I said, for example, this is what happens in a healthy situation. This is a goblet cell, so the mucus layer is constituted of mucin glycoproteins that are produced largely by the goblet cells, at least the secreted mucin. The main mucin in the mucus layer is the mucin 2 and is produced by the goblet cells. And here you see the two layers of the mucus, one that is devoid of microorganisms and the other one more external, where the microorganisms are found. In a situation of disease, like for example ulcerative colitis, which we will talk about more extensively in one of the next lectures, in ulcerative colitis it may happen that the mucus layer is not continuous. So if it is not continuous, it allows for the microorganisms in the lumen to now come into contact with the epithelial cells, which contributes to the inflammatory reactions and response that characterizes this form of inflammatory bowel disease. Okay, so that table there shows which proteins are found in this mucus layer. Obviously, by no means you have to remember all these proteins. I just wanted you to give you an idea of which proteins are found in this mucus layer, and that many of them are mucins. Right? So the mucins in the intestinal tract are very important for so many things. So let's see a few. So the family of mucins, it's about 20 proteins it contains, right? And these are the ones that are expressed along the intestinal tract. These are the mucin genes and their level of expression from higher to lower along different regions of the intestinal tract, gastrointestinal tract, from stomach to colon. Okay? And underneath is a representation, a schematic of what the mucus layer looks like. This was done in the rat, from a rat samples. So showing the difference between the different regions. So in the stomach and in the colon, it's a continuous layer, while on the small intestine, see, this green is the mucus layer because of the different anatomy, right, in the small intestine. So mucins are heavily glycosylated proteins, and they contain essentially the two type of sugar linkages: the O-linked, where the sugar chain is linked to a threonine or a serine, and the N-linked sugar chains. But most of them are O-linked. Okay, so the structures, you don't have to remember the structure, but I want to show you this because it will help you understand slides that will come next in this course. So you don't have to remember the sequence of the sugar moieties in the glycosylation structures of the mucins. But I want you to know what they consist of, because it gives you an idea, because later this information will be needed. So the O-linked, which is the majority of the glycosylation of mucins, right? They all start with a GalNAc, an N-acetyl galactosamine, which then the chain continues until, with all different sugar moieties that are shown there, until it's finished, like the terminal sugar structure are either a sialic acid or a fucose. Okay? When a sialic acid or a fucose are added to the growing glycan chain, no more extension can happen. Okay? So this is the same, these are the same structures that we will find on the human milk oligosaccharides when we will talk about those, I think next week, and also during the second part of the course in Dr. Vansma lecture. So I'm just telling you this so that you start making some connections. So those fucose and those sialic acids are very important for the colonization of the bacteria. So these are terminal sugar moieties on the O-glycan attached chain, sugars of the mucin proteins. Now think that we have an intestinal tract with our intestinal monolayer of intestinal epithelial cells that is covered with this mucus layer, which is rich of glycan, of carbohydrates, basically, right? So one function is just physical of these carbohydrates, because they entrap some of the water, they give the viscous character to the mucus layer so that it can protect, and it can also can protect from bacteria and pathogen accessing the intestinal cells. But these sugars that are found there exposed on these proteins that we produce, right? Because the mucins are produced by our intestinal cells. Now these sugars, especially those in the outer mucus layer, are exposed to the microbiota, right? And in fact, they can constitute an energy source for that microbiota. So those, some of the bacteria in the microbiota possess glycan hydrolases. So enzymes that are able to degrade these sugars so that they can use them as energy source. This is particularly true in the intestine of an infant, because the infant is only receiving milk, either breast milk or formula, but it's not receiving complex polysaccharides like we can find in a solid diet, right? So those microorganisms in the intestinal tract of the infant, they need to feed on something, right? So they feed on the human milk oligosaccharides, but they also feed on the sugar structures that are derived from the host-produced mucins. And so this is an example. Obviously, you don't have to remember all these enzymes and which bacteria express them, but these are examples of enzymes that are able to degrade those sugars and which microorganisms express them. So which microorganisms have that potential. So in some cases, the ability to degrade mucin glycans is beneficial, right? Because so the microbiota is nourished, for example, in a baby. But if it's too much, like always has to be kept under control, because we don't want an over-degradation of the mucus layer, because then the mucus layer becomes too thin and cannot be protective anymore, right? So there are some disease conditions where there is overconsumption of the mucus layer, maybe because the microorganisms are starved and the host, I'm talking about an adult now, the host is not ingesting a diet that is supportive of the microbiota, and the microbiota has to find their carbohydrate source somewhere, right? So they may revert to use host-derived glycans if we don't provide with the diet. So there is a microorganism that is called Akkermansia mucinifera. So this one, you will have to remember this name because it will come up again. So mucinifera, the name means mucin, and it's from Latin. It means that it likes mucins, okay? So it was isolated in 2004. So not, maybe to you seems like a long time, but it's not a long time, 2004. And can represent up to 5% of the total load of bacteria in the gut. So it can be like a lot of Akkermansia mucinifera there. Now this one is specialized in utilizing mucins. So it has up to 61 curative mucin degrading enzymes. And so keep this in mind, because later on, when we will talk about probiotics and postbiotics, these bacteria will come up again, because recently, like 2 years ago and last year, there were papers. It was found like a potential beneficial bacterium in the context of the metabolic syndrome. So it will come up again, this Akkermansia. So what else does this mucus layer do? So we said it protects, so physical barrier, so it's part of the intestinal barrier, particularly it's the physical part of the barrier that physically covers the epithelium, protects from the pathogen to access it. It provides energy source to the microorganisms, and also it acts as a scaffold for the gut bacteria. So some bacteria may attach to food particles in the lumen, some others are entrapped in the outer mucus layer, some others attach to it either specifically or non-specifically, so that it acts as a scaffold for them. So why most of the times we have produced data from stools, and that therefore, I don't misunderstand me, like I have nothing against studying stools, and I do this myself in my studies, right? In my lab there is stools everywhere. So, but just keep in mind, so they're not a proxy for what happens in the ileum, and also they are not a proxy for what is the mucus-associated microbiota, right? Because there is that microbiota that is in the lumen, or that detaches from the mucus and then is found in the lumen and that passes through, and there is also a microbiota that is more adherent to the mucus layer, the composition of which may differ from the one that is found like more towards the lumen, right? So again, there are studies that will focus only on the mucus-associated microorganisms. And those are probably the ones that may be more directly involved with our intestinal cells and exchange of communication. I'm saying maybe because we don't know, right? The communication with receptor mediated, I mean. Obviously, if it's metabolite mediated, then the metabolites can be found in the lumen as well. So, but keep in mind that the bacteria that are attached and adhered to the lumen are not necessarily the same. Like the composition of that mucosa-associated microbiome is not necessarily the same of the one that is more luminal. So this microbiome, we said, right? It has lots of beneficial function. Largely can be largely categorized in 3 groups. So nutrient processing and energy homeostasis. So they help us metabolize dietary components, and they play a role in energy homeostasis. They contribute to the intestinal barrier. So I spoke about the mucus layer as a part of the barrier. The microbiota is a part of the barrier itself, because the microbiota itself can be considered either a physical barrier, like the one in the mouth, if there is a layer of bifidobacteria, or commensals, doesn't have to be bifidobacteria, commensals that is situated covering the intestinal epithelium, then just physical hindrance and the pathogens find it more difficult to control. In fact, a consequence of excessive use of antibiotics can be infection, because excessive use of antibiotics may detrimentally affect our own microbiota and make our intestine more susceptible to infection that could have been kept under control by the microbiota, right? But also this microbiota can produce substances, like we saw earlier for other microbial ecosystems. Like it may produce substances like bacteriocins, for example, other metabolites, even the short-chain fatty acids, lower the pH locally where they are produced, and which keeps pathogens under control. So this is another function of the microbiota. And then, as we will see next week, it regulates the development of the intestine. So it is said that it instructs our immune system, so the development it drives. I cannot say it's the only driver, of course, but it helps driving the development of the immune system in the infant, and also keeps homeostasis, helps the host to keep homeostasis throughout life. So 3 broad functions of the gut microbiome. So the intestinal development, we will see this next week. The contribution to the intestinal barrier, we'll see bits of this across multiple lectures. Obviously, Dr. Philpott will look at this from the immune perspective, but then we'll touch on this in many different lectures. And nutrient processing and energy homeostasis as well. But generally, for today, I wanted to give you some fundamentals on this nutrient processing and energy homeostasis and what these microbiota do. So it participates in carbohydrate metabolism and the hydrolysis of complex sugars, for which we do not have the enzymes for doing this, right? So, for example, fiber, we wouldn't be able to digest without the microbiota, right? It digests proteins, which, yes, it's true that most of protein digestion happens in the small intestine, right? But there is still a certain amount of proteins that reach the colon and then are metabolized by the bacteria. Contributes to lipid metabolism, vitamin synthesis, mineral metabolism, so absorption of calcium, for example, and xenobiotic detoxification. So all of these things we will discuss in the various lectures, but just some basics today, at least for the carbohydrate metabolism, right? It's really critically important that we understand how we cannot degrade complex polysaccharides. But on the other hand, as we saw for the mucins, right, this microbiota possesses because you have to think of the microbiota obviously as a whole. right? So the many different microorganisms contribute specific hydrolases, glycan hydrolases and functional capacity for the degradation of these complex polysaccharides. Right, so these are undigested by us, but are digested by the microbiota so that they can be fully utilized and also generate energy and also generate other metabolites that then are useful for other cellular reactions. And then ultimately resulting, for example, in the production of the short-chain fatty acids. You will be tired of hearing me talking about short-chain fatty acids in this course. So for example, I was saying earlier to one of you, I don't remember which one, so the short-chain fatty acid, remember the three that we spoke about in the first week? So for example, the butyrate, right, is the main energy source for the colonocytes, right? And it is derived from fermentation of fiber that only the microbiota can do and produce the energy, the main energy source of our colonocytes, our intestinal cells, epithelial cells in the colon. So this is really important. So, and again, obviously I don't expect you to learn this table, but I want you to give an idea of how many different hydrolases these microorganisms have, right? And how many different glycan linkages they are able to digest. Right? So one of the main digester is the Bacteroides theta iota omicron. I'm sorry for this name. I did not name this bacterium, but this is how it's called. So it's one of the main ones in terms of glycan degradation, and it's also present in a relatively high percentage across many different people, right? So look, it has a 100, at least based on this paper, 172 different genes for the degradation of glycans, as opposed to E. coli, who has 18, Bifidobacterium longum, NCC 2705, which is a probiotic, that has 39, Clostridium perfringens, which is a pathogen, 29, and Enterococcus fecalis, 21, right? 172. So this belongs to the phylum of the Bacteroidetes. So it's a main degrader of glycans, okay? So what happens, I know that you kind of can put this together with what we saw in the first week, right? But this now specifically for the intestinal tract. So I'm going to repeat a few things, but that's okay. So that's the dietary input in terms of complex polysaccharide that we ingest, right? So they are degraded by the members of the microbiota that in concert act together to digest these complex molecules, right? So they do all that we saw last week, 2 weeks ago, and then they produce this end product that you must know. The short-chain fatty acids, butyrate, propionate, acetate, and the gases, carbon dioxide and hydrogen, right? So these must be remembered. Now, the short-chain fatty acids have different fates depending on which one. So the butyrate, as I said, is the main source of energy for the colonocyte, so it stays in the colon, right? There is very little butyrate in the circulation. There is still some, but most of it stays in the colon. The other two, the propionate and the acetate, go to the portal vein, right? And therefore they reach the liver and then they are in the circulation. Now, what happened to the gases? You know this by now, right? A little bit at least. So they need, now we have a large, let's say large, if there is a lot of fermentation going on because we have eaten a lot of fiber, then we have a large production of gases that are available, especially the hydrogen, for a reduction type of reaction, right? So there are members of the microbiota that are called hydrogenotrophs, right? Let me see if I have this written in the next slide. Yes, the gut hydrogenotrophs. So what do? They use this hydrogen that has been produced by the saccharolytic microorganisms, right? So now we have this hydrogen that these other ones produce. And, well, first of all, these gases can be eliminated through flatulence, for example, right, or in the breath. But some of them can also be used by other members of the microbiota, like the hydrogenotrophs, sulfate-reducing bacteria, acetogens, and methanogens, to produce other metabolites. So the one that we are most interested in are the methanogens that we saw a few times now, right? So what do? There are different methanogens, remember that slide that we saw earlier, and there are different substrates that they can use to reduce this hydrogen, to reduce different substrates. So if they reduce the CO2, like the Methanobrevibacter smithii, they produce methane. But they can also use trimethylamine, TMA, DMA, MMA with the hydrogen and produce other compounds, right? So for example, the TMA, we'll discuss this in the last lecture with Dr. Jenkins, but is the one that is important in the context of the TMAO and cardiovascular disease, right? So the TMA can be reduced by the archaea using the hydrogen that is produced by the fermentation of the other bacteria that use the fiber. So yeah, so that's like a large scale example of what happens and how the different metabolites from the carbohydrates can be utilized. For the proteins, so I said that most of the proteins are digested in small intestine, right? But there can be like 6 to 15 grams that can leak into the colon. So when they go into the colon, these proteins become available to the gut microbiota that can metabolize these compounds. And they can produce many, many different metabolites. Now, the metabolites of fiber fermentation are basically beneficial, right? It's those short-chain fatty acids, it's those gases that mostly are utilized in a manner that is beneficial to us. In the case of the protein, what they produce from the protein metabolism is not always beneficial. So what we can obtain, we can still obtain short-chain fatty acids and gases, but we can also obtain branched-chain fatty acids, phenols and indoles, ammonia. They can deaminate the proteins, the peptides, amino acids, so amines, and so on and so forth. So these substances can be toxic to the cells, right? So, and many of these substances have been linked to colorectal cancer or other types of cancer. So when we will have the lecture on colon cancer, there will be the protein digestion will be discussed in depth to understand how the microbiota may not always be beneficial, in fact, and then it all depends on the dietary inputs, right, and the relative abundance of fiber versus protein versus what we ingest. But from proteins, there could be toxic compounds that are produced, right? Generally speaking, the fermentation of fiber happens more proximally in the large intestine, while the protein happens more distally in the large intestine. Yeah, and that's it for today. So, any other questions? No. So I'm sorry for the thing with the discussion board on the puertos. I don't know what happened. I will check it, and hopefully I can fix it.
큰일 났어.